This poster presentation is called Investigating the Role of the Gut Microbiome as a Mediator for Radiation Therapy-Induced Bowel Injury, and my name is Alex Jameus. Before beginning my poster, I would just like to add that I have nothing to disclose, no affiliations with any for-profit or not-for-profit organizations, and the financial support for this project has been received through NSERC, CAHR, and NHRC funding agencies. Radiation therapy is a popular form of cancer treatment that over half of all cancer patients will undergo at some point in their treatment journey. A large issue with radiation therapy is that the radiation passes through normal healthy tissue in its path to the tumor. The tissue of the gut happens to be some of the most radiosensitive in the body, meaning that it's not very tolerant to the radiation. This leads to a long list of side effects that can negatively affect patient quality of life. Medically, this can be called GIARS. An important part of maintaining a healthy gut is the gut microbiome, which refers to all of the organisms living in the gut, mainly bacteria. Many studies point to the bidirectional role of the gut bacteria in the management of cancer, as well as the occurrence of treatment-induced side effects. For this study, 40 male C57 mice were irradiated at 16 weeks of age in doses of 0, 0.1, 0.25, 0.5, and 3 gray. At each dose, sections of the ileum were stained using hematoxylin and eosin staining and analyzed to determine the histological effects of irradiation on the gut. We looked at villi height, crypt depth, the number of goblet cells within the crypts, and goblet cell size. Irradiation had no significant impact on ileal villi height, however, 3 gray significantly reduced crypt depth and increased the goblet cell area, but decreased the overall number of goblet cells. There was no impact seen by the 3 lower doses of radiation. Total genomic DNA was extracted from frozen sections of the gastrointestinal tract, as well as fecal samples, and then sent out for 16S metagenomic analysis to determine the effects of irradiation on the gut microbiome of the mice, which shares roughly 90% similarity to humans. In Figure 2, we show the 2 dominant phyla of the gastrointestinal tract. This is the same for both humans and mice, and they are firmicutes and bacteroidetes. At this uppermost level, we saw no significant changes in bacterial composition. Figure 3 shows 2 order of bacteria within the firmicutes phylum and shows that various doses of radiation produce significant changes in the small bowel in both cases. Next, Figure 4 shows 2 families of bacteria within the Bacteroidetes phylum were significantly altered following radiation exposure, particularly within the colon and feces at 3 gray. To conclude, this study provides insight into how ionizing radiation exposure can induce acute alterations across various taxonomic ranks in the gut microbiome of mice. We did see alterations in the gut following radiation, both histologically and compositionally. While the villi were unaffected, there was a whole crypt effect following radiation exposure. Compositionally, within the 2 dominant phyla, there were no direct changes. However, as we descended down taxonomic ranks, we saw changes to the small bowel within the Firmicutes phylum, and in the Bacteroidetes phylum, we mostly see changes within the colon and feces. We believe there to be a potential use for this data as a biomarker for monitoring radiation therapy patients or indicating targets for mitigation strategies. This means that we might be able to solve or at least lessen problems before they occur if we have a way to indicate potential issues down the line, overall leading to better management of side effects and disease control.